Good evening to everyone. It is my great honor and privilege to present before you today as the keynote speaker in this 7th edition of Global Congress in Plant Biology. I would like to begin my presentation by thanking the organizer, this Congress, for inviting me to speak as a keynote speaker. And I think there is, okay. Please go to first slide. Yeah. Today I'm going to discuss about, it is a review and also our research in working in our laboratory, antimycobacterial activity of plant molecules from traditional medicinal plant. So traditional medicinal plants, everyone, it is very important because most of the people are utilizing traditional drugs to cure their various ailments. How can I go for next slide? Yes. Okay. Before that, I would like to talk about tuberculosis. Why we are looking the molecules for the control of tuberculosis from the traditional medicines. Mycobacterium tuberculosis is very ancient origin. It has survived 70,000 years of, and currently, in fact, nearly 2 billion people worldwide. Definitely, it is creating very dangerous situation in the world. Particularly, developing countries, it is a major health problem. 30% of the world populations are infected with Mycobacterium tuberculosis. WHO estimated more than 10 million people fall in with the TB, and 1 million people were died every year. And incidence rates are reported in all countries and age group. The disease affect mostly 57% women, account for 32% of the cases. So about 1/4 of the world population has TB infection. See, everywhere, particularly during the COVID situations, the rate was decreased because of due to there is no proper diagnosis. That is why the result was showed. Low rate of infection rate, but prevalence of the TB. But when we see the mortality, it was increased. Next. You see, World Health Organization recently reported that around 1.6 million people died from the TB in 2021 year. And also 2021, it was estimated 10.6 million people fell ill with tuberculosis worldwide. And 2021, 1.2 million children also fell with TB globally. So this is a very dangerous, and its prevalence day by day increasing throughout the world. In 2021, report said that 30% of high TB burden countries are accounted for 87% of the new TB cases. And why? Because multi-drug resistant TB, Mycobacterium tuberculosis, are arised everywhere and remains public health crisis and health security threat. Next. This slide shows the current data of the tuberculosis in world and India. Because, see the rate up to 2012 to 2021 reported by the Trend Pediatric TB notifications showed that it was increased and around, but 2020, it showed low rate of TB. Why? Because it is a COVID situation. So there is no proper diagnosis. That is why it showed results very low. Next. Globally, TB mortalities showed that, and you see, you can able to see the most of the countries are from the African countries. African countries, around more than 40% are affected. Particularly in India, you see 20 to 39% mortality were occurred. Next. So how it's happened? What is the risk factor of the TB? How it is coming here. And the people who have poverty with HIV infection and homelessness people, some of the people are having substances. I mean, this, for example, nicotine or some other narcotic things, and taking medication that weakness the immune system, whoever, and whoever have kidney disease and diabetes and organ transplants, these people are risk factor to affect the tuberculosis. Next. This slide shows the transmission of tuberculosis. Transmission of tuberculosis is nothing but it is an infection. It is airborne microbes. When source of the microbes, who has the patients, how the source of the case is from the lungs. Automatically when she has sneezing, the bacteria will come out from the mouth and it will spread on the aerosolization, release the viable airborne bacillus, and then it will easily, who are having low immunity and the risk, what I said previously, risk of factors, they will immediately affect. So this is the transmission of tuberculosis. Next. So currently, anti-tubercular agent, this is first anti-tubercular agents are discovered by the scientist Leodol. Entomology came in 1961 by Leodol laboratories. Then fluoroquinones, newer macrolides, and rifampicin and rifampicin. These are the antimycobacterial are derived first time. The currently still date rifampicin is one of the active antimicrobial drug. Next slide. So currently used drugs recommended by the WHO. These are the drugs. There is a different categories. The group A, it's mostly related to fluoroquinones, and the group B and the second line injectable agents, and group C, other core second line agents. So these are the currently used antimicrobacterial drugs which are recommended by the WHO. Next. So this is slide shows the innovation gap in TB drug discovery. You see, in 1940 onwards and 2010, from the 1960 to 2000, there were no any new drugs discovered for the tuberculosis management, because I think currently there are several drug-resistant mycobacterial tuberculosis are arising, and it has created several issues. But the scientists are still there. Whatever the already used drugs, it was continuously modified with several groups, and it may be have significant activity against the tuberculosis. And that's why our research also, we are looking for novel molecules against the mycobacterium tuberculosis, which has low side effect and with using some edible plant or traditionally used plant. Next slide. Before that, I would like to discuss about what is the traditional medicines. So traditional medicine is nothing but WHO already defined. It is knowledge, skill, and practices by the culture of the people, or traditional people, or tribal people. For example, if you have any ideas or theories, it has to be used for the management of your health. For example, diagnosis of prevention, and improvement. These are all defined as a treatment of physical and mental illness. It is called as traditional medicines. Most of them, traditional medicines are derived from the plants. Next. There are different categories. We can be able to see, category one is traditional medicine that are prepared by the traditional healer and practitioner, and for using for the individual patient. And category two, traditional medicine currently used in the community as composed as a crude raw plant material. That is I think from the tribal people. And category three, standardize the plant extract prepared in advance in supported by the scientific research. This is the category we are doing. And next four, isolated pure compound molecules from the traditional medicines following with the scientific research. In traditional medicine system in India, this is the following system are followed. For example, Ayurveda, Naturopathy, Yoga, Unani, Homeopathy, Siddha. These are the traditional medicine system very effectively running in India. Next slide. So what are the roles in the, medicinal plant, particularly traditional medicinal plant? Possess different biological properties. For example, it has thousands of secondary metabolites. Each plant have thousands of secondary metabolites. It is called phytoconstituent. And there is alkaloids, flavonoids, triterpenoids, and anthraquinone, saponin, terpenoids. So different kind of the class of compound is available in the medicinal plant, and all are having some specific biological properties. That is called different biological properties. For example, anti-hyperlipidemic, anti-spasmodic, antimicrobial activity, anti-malarial activity, antiviral activity, wound healing activity, analgesic and anti-pellet aggregation activity, anti-cancer, anti-accident, and anti-inflammatory, finally anti-diabetic activity. Even though these activities are expressed, nowadays people are using traditional medicinal plant as a insecticidal activity also. This next. So medicinal plant, how it is used to isolate the drug for the tuberculosis. Globally, traditional medicines, it is playing vital contribution to health management, particularly in African countries. There are several traditional medicines are reported by the traditional people. In India, it is a 12th mega biodiversity country. We have several traditional medicine product which is currently using for the human healthcare management. Among the 435,000 plant species reported worldwide, and estimated nearly 70,000 species plants are used for medicinal purpose. It is increasing efficiency to develop the new drug and raising concern about the side effect and of modern medicine. And research are exploring novel antimicrobial compounds that have lesser side effect due to the development of multi-resistant TB and the severe side effect of the synthetic drugs used for the treatment. The recent ethnopharmacology survey showed that medical practice related to TB treatment is increased by the natural healers are still alive, and they are using now different countries. For example, South Africa, Uganda, Ghana, Malaysia, and India. Next. Next slide. This slide shows the medicinal plant used for the TB management. You see, there is around, different kind of families or plant which is having nearly, thousand of the 2,000 something. For example, Asteraceae family, it has more than 25% of the plants are used for the tuberculosis management. And for example, here, the Psychobilaceae, it is the lowest. I mean, this family, more than 5% plant are using for the TB management. So, I mean, most of the research are trying to explore the novel molecule from the traditional medicinal plant for managing the tuberculosis. Next slide. So how the plant-based food products are used for the antimicrobial therapy. For example, you see, usually every day we will have pepper and ginger, curcumin, and I mean clove. These are all having some biological property which is highly protecting the hepatotoxicity, I mean against the hepatotoxicity, and protect the liver and lungs. Lung is source of the mycobacterium pathogenicity occurring in the lungs. It will affect. So these are what I said. These are the, I mean, plant-based food products currently very much useful to control of the Mycobacterium tuberculosis. Next slide. Please, next slide. Yeah. This slide shows the, I mean, for example, many synthetic drugs currently, I mean, previously and currently are using, which is showed highly toxic to the liver, and it has created some drug metabolic intermediates and also produce hepatotoxins. It will affect the liver. The liver will damage by the cirrhosis. It means it have sometime it will have produce, I mean, fatty liver disease also. And because of there will have changes in oxidative stress and immunodysfunctions. And plus, next one, the anti-TB drug plus with the traditional, I mean, medicinal plant. With combine these drug, when we have the phytochemical stimulate the liver enzyme and control the produce the hepatotoxin and product the hepatoproductive, and the liver will be health. And also it will inhibit the microsomal drug and metabolize the enzyme and reduce, I mean, plant chemicals. Phytoconstituents will reduce the oxidative stress and increase the antioxidants. So wherever the plant have antioxidants, it will useful to the, I mean, reduce the free radicals. Next slide. So this slide shows the most frequently used edible plants recommended for the TB management. And you see there is several medicinal plant used for the TB management. For example, particularly Allium ciba, it is anion, and aloe vera. Next, I mean, Anonas comosova, Cassia fistula, it is one of the ornamental plant. Centella asiatica is the edible plant. Citrus microscope, it is edible. So most of the plant, for example, Cinnamon cambophora, and here Asimum sanctum. So most of the plants are edible. And why we are choosing this plant for management of TB and to develop the new drug, because edible plants will have that will will not have any side effect and toxic material. So that is why our research are only from the traditional medicinal plant. Next slide. There are several researchers. They have continuously doing the research using the medicinal plant and reported different kind of molecules, plant molecule, which is highly inhibit the, Mycobacterium tuberculosis and with the lowest concentration. Here you see the Blading et al. 2019, he has reported Singer officinalis rhizome. He found essential oil and which is highly inhibit the mycobacterial tuberculosis at lowest concentration from the 31.25 microgram to 250 microgram. It is very good, effective. And here, one of the compound, 10-gingerol, isolated from the methylene chloride extract. The same plant, it's inhibited the Mycobacterium avium. It is just for 20 microgram per ml concentration. And here, stigmasterol, this is one of the compound which is isolated from the hexane extract from the menthol citrifolia. So it is highly sought activity. The next slide shows the list of common polyherbal formulation by the Ayurveda system in India. Ayurveda, Unani, and Siddha currently used as a product for the management of tuberculosis. For example, the asthma condition, people, they are using Ayurveda system. The drug name is, Himalaya company. They produce different type of the tablets and some, formulation. For example, Bristol tablets, which is highly cure the asthma, and Curcuma longa. It is one of the, it is the common name is turmeric. It is highly used to control the TB. And these are the, Asimum sanctum, Adathoda vasica. It is all our formulations. And from the Unani also, you see, so many formulations, Ravalbia serpentina, Acorus calamus. These are the plant have used as a formulation and for control of the TB. And Siddha, there is using for the Tipili, Piper, Langam, and Gingiber, and other so many Solanum nigrum plants, gingerly oil. Solanum nigrum, it is one of the edible vegetables, small vegetables, widely used for the, inflammatory activity also reported. So these are the Ayurvedic formulations or prepared by the Indian system, the traditional system currently used for the tuberculosis management. Next slide. So this slide also shows the Indian medicinal plants used for the tuberculosis in India, and particularly where you see many of the plants are edible. Centrala asiatica, Coccinia grandis, it is one of the vegetables. Next slide. This slide also shows the, several researchers are reported from the traditional medicinal plant and control the tuberculosis. For example, Acanthus purpurans roots are used to treat TB and related disease. In Uganda, he has reported. And Mangifera indica, it is mango tree. You see, stem bark used to treat the TB and related disease. And in Uganda. And for examples, in Nigeria, Elysis guinesis and oil soup is used to do the treatment of TB. It is a traditional drug. Next slide. This also, so many of the researchers, they are isolated the compound and tested against the Mycobacterium activity and Mycobacterium tuberculosis, normal strain and as well as resistant strain, which is so highly sensitive. For example, urosilic acid, cucurbitacin, these are the compound or isolated from the citrulose cholesynthesis. And for Lipia javanica, it is one of the important medicinal plant. And eicosalic acid and dacagitones, epoxide, mycinone, these are the compound which is highly inhibited the Mycobacterium tuberculosis. They have reported in the 2011. Next slide. Now I am coming to, in our experience in our laboratory. Our laboratory is a multidisciplinary research. We have different divisions, division of biochemistry and ethnopharmacology, division of microbiology, division of, biodiversity, division of plant biology and biotechnology. We have run more than 2 projects which were funded by the, CSCR and another one ICMR. So these are the related publication we have published for antimycobacterial activity of compounds isolated from the traditional medicine. Next slide. So isolation of the active principle from the traditional medicinal plant. So these are the contained, traditional. First, we have to get the traditional knowledge. That is why two of my research scholars, they went to, met the tribal people and got the knowledge, and then we will go to the field, collection the plant. Then plant, before we choose the plant, we get the knowledge from the tribal people, and the plant has to be identified with the help of the botanist. And then the sample collection, during the sample collection, we should definitely to take care of the following factors. For example, its plant should not have any insect damage, and particularly should not collect the young leaves, because we are looking for the secondary metabolites. So most of the secondary, plant physiology, the secondary metabolites will produce mostly available in the mature plant. So we should collect before we have some factors, and then collect the plant. We have to bring to the laboratory, and there should be thorough wash with fresh water, and then we have to go for the extraction preparation, then biological assay and the bioassay gated fractionation, then purification and characterization of the molecules. So these are the following things we are going to see. Next slide. So this is the field we have collected with my research scholar. So for example, one of the plant we collected, during the plant collection, we should have some safety measures, and we should have gloves and mask, because there is some insect or something will be attacked. So we have to take care of ourselves. Next slide. Then after identification of the plant, it was completely dried with room temperature only, because when we have sunlight, dryness means the ultraviolet rays will affect the active compound. Maybe it will be degrades. So mostly we will have in the dark room with room temperature. So after bring it to the plant should be chopped with small pieces and then ground with a manual mixing as a coarse powder, not not like a powder form, just for coarse powder. Next slide. This is a procedure we are following in our laboratory to extraction of the medicinal plant. As I said earlier, this following method we have to do, and finally we have 1 kg dried powder, which has to be poured in the aspirator bottle or any bigger, conical flask. Here we are going to use, we were used 1:3 ratio, weight by volume. For example, 1 kg powder, we have to use 3 liters of solvents. So we will, this is we are using here sequential extraction method. Mostly we are using here 3 different solvents, polar solvent, hexane, ethyl acetate, and methanol. Hexane is a low polar, ethyl acetate is a medium polar, methanol is a high polar. We can, at a time, we can test 3 different polar components against the appropriate bioassay. So after that, then it has to be kept for 72 hours. After 72 hours, it filter and concentrate with the vacuum rotary evaporator, and then we will finally get crude extract. Then crude extract directly to test for bioassay. Next slide. So after done biological assay, we will do for the column chromatography. This is one of our work has done in our laboratory, and the crude extracts will pack the column chromatography, with the help of, different organic solvents. Basically, here we will use the column will pack the low polar solvent hexane. Then we will increase the polarity in the different bases, hexane ethyl acetate and hexane chloroform ethyl acetate methanol, like that. Different polar ratio. Then after all the fractions will combined, and we will test the TLC, and is that you can be able to see their different components. And then if there is, 2 or 3 compounds presence, again we will go for the subtraction, and we will isolate the compound and then concentrate the fractions. Finally, we'll get the pure compound. The compound will go for the structure elucidation by the spectral analysis. Next slide. So by the way, we are isolating many molecules from our laboratory. But first one, the plant Adatura vasica, which is a highly traditional medicine used by the tribal people in different countries, which is, more than 2000 years. It is used by the traditional people for as a medicines, and it is for particularly the management of tuberculosis. So this, based on these information we have collected, and we have isolated the compound from our laboratory. Next slide. So this is a compound, the Adatura vasica, we have collected and dried, and then 1 kg plant material extraction with alcohol. Here we are looking only alkaloids. No need to, because alkaloids is a major components in presence in the Adatura vasica. Previously many people are reported vasicin is the major alkaloids. It is used to cure the tuberculosis. So we wanted to isolate that compound and modify some of the group. This compound we use isolated by this method. Extraction with alcohol, and then this 60 gram extract with acidify with 5% of acetic acid, and then ammonia, and then extracted with these solvents mixed with a liquid condition. Then we extracted with the chloroform. Finally, we got it there. Crude alkaloids, vasicin. It was compared with the standard vasicin. Next slide. So this then crude alkaloids, which was packed the column chromatography and isolated nearly 28 fractions. These fractions we found particularly 7 to 14 fractions. It was run by chloroform methanol. The mobile phase solvent system is chloroform methanol. These fractions and showed some pure component. Next slide. This is the TLC, thin layer chromatography. We have run for all the plates, fractions. You see, able to see here, 7 to 10, and particularly 13, there was single spot. So because crude alkaloids finally purified as a vasicin, and this was then combined and got it a pure compound. Next slide. You see, so among the 8, what I said, 7 to 14 fractions combined, we got it, pure compound. This compound, around 1.7 gram, we got it, pure alkaloids. Next slide. So next, we want to acetylation of the vasicin, and 1.7 gram vasicins, and use the acetic acid and added 5 ml of pyridines, just mixed it together and kept for the overnight, and finally poured into the crushed ice, and then aqueous solution based with the ammonia up to 10 pH. Then it was, you can see the precipitated vasicin acetate. Then these compounds extracted with the chloroform solution, and then finally concentrated by the vacuum rotary evaporator. Then it was tested. You are able to see the slide shows this is the standard vasicin. Then the vasicin, after reaction with this method, you can able to see this is unreacted vasicin. This is a vasicin acetate. So finally, we got it in novel compound, vasicin acetate. Next slide. Next slide, please. Yeah. This is compound, vasicin acetate. Showed, same what I said. Then it was submitted to the test against the tuberculosis. It's inhibited the around, sensitive strain for even normal and multi-drug strain. Showed 50 microgram per ml. It's inhibited the Mycobacterium tuberculosis. It is isolated based on the traditional knowledge. Next slide. This is the compound we have reported in the paper for antimicrobial, but antimycobacterial activity still it is under process. Vasicin acetate. Next slide. This is the next one plant, Cassia fistula. It is a common ornamental plant, highly medicinal value. Is used traditionally Ayurveda and Chinese traditional medicine, and used for the antimicrobial, anti-inflammatory, and anti-diabetic activities. It has been used to treat the tuberculosis, rheumatism, leprosy, and syphilis, and skin disease. This is a traditional knowledge. We got it, and we went for the isolation of the compound. Next slide. This is the method we have isolated. The same thing we followed the column chromatography, and around we have collected 117 fraction and combined together by the TLC, and from the 25 fraction we combined. Then from the we have isolated two compounds. One is from the fraction 9, another one is 18. When we see the TLC, there was single spot showed. Then it was this compound from the fraction single 9 showed a single spot. It is showed crystal. The after obtained crystal directly went for the crystallography studies, and we got the structure, the compound name. Next, the compound from the fraction 18, this showed single spot on TLC. We subjected to the spectral analysis, we got it the compound 2. This is the first compound. One is a benzoic acid, and the compound 1 is a rain. It is anthraquinone. Next slide. So this is the slide we run for the TLC, all the fractions, all the combined 24 fraction. You see here the fraction, 18 showed single spot. Other fractions are having some impurities. So only the fraction 18 showed single spot on TLC. It is considered as a pure compound, which is only the compound or pure compound. We can submit to the spectral analysis for the structural elucidation. Next slide. So after the spectral analysis, we conclude the structure is anthraquinone. It is a common name is rain 1,8-dihydroxy anthraquinone 3-carboxylic acid. The molecular formula is C15H8O6. So this compound we have tested. It showed good activity against the antimicrobial, and also it was tested anti-cancer activity. Next slide. So we have isolated several compounds only based on the traditional. So you see the Emplia ripes, it is a fruit, seeds. Its a common name is a vedanga. It is the pot used for the seed. Traditionally used for this compound diuretic, powerful, anthelmintic, and anti-fertility, anti-cancer. From this we have isolated compound is empline, which is highly showed anti-diabetic activity, as well as we have submitted to the mycobacterium activity. Next slide. So Plumbago geylanica, it is one of the plant we isolated the plumbagin, which is highly active compound. Next slide. Next slide. This is a Rheum emodi. We have isolated the compound from this plant, trisulfenol, which is based on the traditional uses, which is showed. These are the biological properties. Next slide. Elephant scaber. This is a highly medicinal value, which is currently using the people or management of diabetics, because which is highly reduce the blood glucose level. And in our laboratory also we have reported many compounds and used as a diabetic agent in animal model. And this also, I mean, used for the traditionally management of tuberculosis. Based on this, we have went for the column chromatography and isolated lupeal compound, which is submitted to the mycobacterial and antimycobacterial activity. Next slide. Acima tetrachondra. This is the one of the traditional medicinal plant. It has different kind of biological properties, and it is used for the asthma. Asthma is related to TB. We have isolated. The compound name is Fridolin. It was reported 2016 by our group. The compound is also submitted to the antimycobacterial activity. Next slide. And Puncomia pinnata. This is a common, I mean, everywhere we can see this plant in India. This plant how also traditionally used for the cough. This is the one word, cough. Based on this, we collected this plant and went for fractionation. Finally, we got it crude carrageen, the major compound, which is also showed highly insecticidal compound. This was submitted to the, I mean, mycobacterial tuberculosis. Next slide. And Tordalia aesthetica, it is one of my PhD work planned. We have isolated several compounds, particularly, I mean, flindresen. This compound is highly inhibit the, I mean, pathogenic bacteria as well as mycobacterial tuberculosis, and also this plant how we isolated another one compound which is highly effective against the diabetics. Next slide. This is a Castor speciosus. This plant is highly used for the diabetics only, but we isolated based on, I mean, our division is multidisciplinary research. So we isolated two of the compound from this plant, costanolyte and erymanthine, which is also submitted to the mycobacterial antimycobacterial tuberculosis, and which is showed good activity. Next slide. So these are what the previous showed compound which were showed good antimycobacterial activity against, particularly for mycobacterial tuberculosis. It is a I mean organism. For example, empelin. I mean less than 16 microgram. I mean plumbagine only 16 microgram inhibit the mycobacterial tuberculosis, and erymanthine inhibit the just for 8 microgram. It is a highly significant activity, significantly inhibit the mycobacterial tuberculosis. And costanolyte 16 microgram, crisaphonol just for 8 microgram, and lupeal 32 microgram, carrageen, I mean fridolin mixer 16 microgram, carrageen 8 microgram. Next slide. And this is the next costanolyte, and some other, we have synthesized some of the inorganic compound from the other lab that are also tested. Here you see, when we compare, rifampicin is used as a control. It is inhibited this multi-drug resistant mycobacterial tuberculosis at 2 microgram. Here you see our compound is the lowest concentration is 16 microgram. So all our isolated compound based on the traditional uses only, we isolated in our laboratory. Next slide. So this is the one of the plant currently used by the traditional people and currently used as a drug, Imburalegium. It is the plant is older than umbellata. There is no any report scientifically isolate any pure compound from this plant for against the tuberculosis. So currently this work is going on in our laboratory to isolate the compound. Next slide. This Tordalia aesthetica, again we are doing one more principle. We are got it, and for which is submitted to the antimycobacterial activity. Next slide. This Alsonia scholaris is that it is used for the tuberculosis management This compound we are going to isolate, this plant we are going to isolate the novel molecules for against tuberculosis. So currently this work is going on in our laboratory. Next slide. Yeah, this is the plant Lipoia javanica and Carica, Ficus carica. This is a Ficus carica. Next slide. So current research work and also going on this plant, we have only look at the, traditional uses, and if there is no any, compound level report, we will take the plant and we will do in our laboratory and we will isolate the molecule. Next slide. So this is the next one, proton on plant yanum. It is this plant is going on in under our lab. Next slide. This is the next one, Curcuma cassia. The microbial activity they reported some only in the extract level. We are going to analyze the molecular level. Next slide. This is the currently Ayurvedic herbs and medicines for tuberculosis. These are the list of the, Viduraipandiyan and Brahmi, Razonam, and Yastin Madhu, Aswakanda. It is one of the very important, traditional medicine to use for the management of tuberculosis. Next slide. So overall, my conclusions from our studies, more plants with their crude extracts showing anti-TB activity. Overall, worldwide, many researchers are trying to explore novel molecules from the traditional medicine to control of the tuberculosis. And the literature said that most of the researchers are, consult with the traditional healer and getting their knowledge to isolate the molecules against the tuberculosis. Our systemic analysis and the medicinal plant can assist the discovery of novel and more efficacy anti-TB drugs. It can be, and whatever isolated our molecule, it can be converted as a product to use for the TB management without side effect. Next slide. These are the other our compound for isolated from our laboratory based on the traditional uses, and some of the molecules we are trying to convert as a product level, and we have collaboration with several companies and two more, medical related, we have some stuck up, and we are continuously trying to convert as a product for against different biological actions, for example, anti-diabetic, anti-cancer, and inflammatory, and my tuberculosis. Next slide. And also not only our institute concentrated, this is our, based on the traditional medicines, we have developed many products for against the mosquito and the insect. This is the moss away. This is one of the plant product, herbal product. And next here the moss repel, moss wave, and this is the stick for the mosquito coils. And these are, and one of this is a ponim. It's highly effective in agriculture field for the control the agriculture pest. These are the products we have found from our laboratory based on the traditional medicines, which is really very interesting work. And we have also got several Indian patents, and as well as some of the compound isolated from the microbes. We are working on actinobacteria. We have got one international patent also. Next slide. And this is our area expertise in our institute. We are working on biodiversity of insect and one of our colleagues, and microbiology and plant biotechnology and phytochemistry, integrated pest management, and environment ecology. This is our institute work is now currently going on in different field. Next slide. So this is the reference I used for the my presentations. Next. Okay. This information I would like to share from here. I was reading this news. India may come up with vaccine against tuberculosis by 2024. It is said by the, FDA Commissioner, Senior IAS official, Steve Campbell. It was, I read in the, newspaper, maybe, they enrolled 12,000 participants and aged 6 years and above of our trial has been completed, and their follow-up will continue till 2024. I hope maybe it will be, India will succeed. It is very proud for us. Next slide. So finally, I thank to the organizer and the participants and the eminent professor, scientist. Thank you very much for listening my presentations. Thank you for all.